The photosynthesis process takes place in the chloroplast of plant cells, where pigments absorb light energy. Chlorophyll, the main pigment in plants, traps light energy, primarily in the blue and red parts of the light spectrum, and uses it to produce chemical energy. These wavelengths are critical for the plant's energy production. While green light is reflected, making plants appear green, by absorbing different parts of the spectrum, plants optimize their ability to capture light energy, which is essential for growth and survival of a plant to get the energy they need to create glucose. So as we can see from the graph, plants primarily absorb violet and red energy, as we can see from chlorophyll A at its peak and chlorophyll B at its peak. On this graph, on the y-axis, is the relative absorption of a plant, or the pigment. On the x-axis is the wavelength, or the light energy. As seen in our first pigment, chlorophyll B, it absorbs a lot of light energy from violet and blue, which are lower wavelengths. So as seen, lower wavelengths equals higher energy. On the other side of the spectrum, we have orange and red, which are 650 and 700. As seen here, from chlorophyll A, it is lower. So higher wavelengths equal lower energy. And in the middle, we can see for all three pigments that green is very low on the relative absorption scale. From this, we can infer that green is reflected, which causes plants to be green.
This is the inside of a chloroplast molecule in the cell. As you can see, the outer shell is the outer membrane, and the inner cell is the inner membrane. The stroma is the fluid-filled space surrounding the thylakoid and in between the inner membrane and grana. This is where the light-dependent reactions or Calvin cycle occur. The thylakoid is a single green circle, or tiny compartments found inside of the chloroplast. The thylakoid is where light-independent reactions occur. Inside of a thylakoid is a lumen.
The overall balanced equation for photosynthesis is 6 carbon dioxide molecules plus 6 water molecules plus sunlight produces glucose and 6 oxygen molecules. Carbon dioxide and water are the reactants, and glucose and oxygen are the products. Carbon dioxide is taken in from the atmosphere through small openings in the leaves and enters the plant cells. Inside the plant, carbon dioxide is used in the Calvin cycle to convert it into glucose. Plants absorb water from the soil through their roots, which transports it to the leaves. Water is used in light reactions to donate electrons and is broken down to form oxygen. Sunlight is the energy source that drives the entire process and is absorbed through the chlorophyll. Glucose is produced in the Calvin cycle and serves as the plant's food source. The glucose molecule is formed during the Calvin cycle. 2 molecules of G3P formed during this cycle are used to generate glucose. Oxygen is a byproduct produced from splitting water molecules in light-dependent reactions. It is then released into the atmosphere.
The light-dependent reactions of photosynthesis occur in the thylakoid membrane between the outer stroma and the thylakoid lumen, which is the interior. In step 1, pigments in photosystem 2 absorb light, which breaks water molecules and excites electrons, giving them energy. Excited electrons leave photosystem 2 and travel down the electron transport chain towards photosystem 1. At the same time, H+ ions are pumped via active transport across the thylakoid membrane into the thylakoid, creating a positive charge inside. They're brought from the lower concentration in the stroma to the higher concentration in the thylakoid lumen, which is active transport and requires energy. In step 3, electrons are re-energized in photosystem 1. The energy inside of the electrons was used in step 2, so they need new energy from the incoming light. In step 4, electrons leave photosystem 1 and travel down the electron transport chain again. The electrons are eventually picked up by the electron carrier NADP+, which becomes NADPH when fused with the electron. In the last step, step 5, H+ ions diffuse out of the thylakoid, passing through ATP synthase, which creates ATP. This is passive transport, as the higher concentration of H+ in the thylakoid lumen goes out into the stroma. The light-independent reactions, or the Calvin cycle, is 4 steps long, and it happens in the stroma because it doesn't need light to start. Carbon fixation, the first step. Each carbon dioxide molecule is added to a 5-carbon molecule, RUBP, by an enzyme RUBISCO or Rubisco. The 6-carbon compound, that after being made, splits into 2 3-carbon compounds. The result is 12 3C molecules called PGA. In the second step, Phosphate groups from ATP and electrons from NADPH are added to make 3 carbon sugars, half the glucose molecule. This energizes the 3-PA. Step 3. 2 of the 3 carbon sugars leave the cycle and are assembled to make organic compounds, glucose, which are stored for later. Because glucose is a 6-carbon glucose, 2 of these makes 1 of them. So, the Calvin cycle, each rotation makes 1 glucose. And then in step 4, the remaining 3 carbon sugars are used to regenerate the 5-carbon molecule, RUBO, from the beginning of the cycle. As an overview of the light-dependent and Calvin cycle reactions, we see that the reactants of the light-dependent reactions are light energy, water, ADP, and NADP+, and the products are ATP, NADPH, and oxygen gas. And then for the Calvin cycle, the reactants are nearly the same, but they are carbon dioxide gas instead of oxygen gas, but they both have ATP and NADPH. So the product of the light-dependent reactions comes over to the Calvin cycle to be used as a reactant, and the products of that are ADP and NADP plus in 1 glucose molecule. And that ADP and NADP plus go back to the light-dependent reactions to be reused like a battery. C4 and CAM plants managing the heat. C4 and CAM plants have adapted to hot environments by storing carbon dioxide as malic acid for later use. They each have their own way of separating the light reactions from the Calvin cycle to avoid photorespiration. The problem of heat. Hot temperatures mean water evaporates from the plant more easily. The plant may close its stomata in the daytime to avoid this. Closed stomata means that no CO2 is entering the plant, and that means there's no oxygen coming out either. If oxygen is allowed to build up near where the Calvin cycle happens, an unwanted process will occur called photorespiration, where oxygen replaces RUBP in the Calvin cycle and resulting in CO2 instead of glucose being formed. So what are the plant solutions to this? They isolate the Calvin cycle away from the light-dependent reactions, where oxygen accumulates in the chloroplast, while stomata are closed in the daytime. Specifically, C4 plants store carbon dioxide for later use so they can keep stomata closed during hot days to minimize water loss. And these plants have a physical isolation, separating in location of the Calvin cycle from oxygen buildup. And as we can see here, in the mesophyll cell, the light-dependent reactions occur, creating malate or malic acid, and then that is used as a CO2 in the Calvin cycle in the bundle sheath cell for C4 photosynthesis. And then in CAM photosynthesis, it's temporal isolation, which means it's separating in time of the Calvin cycle from oxygen buildup. So during the night, it opens the stomata because it's cooler and less arid, and it can absorb the CO2, which then is stored as malate after the light-dependent reactions, or malic acid, and then that's used as CO2 in the Calvin cycle during the day because it doesn't need to have the stomata open. And so the CAM photosynthesis occurs in a change of time, and the C4 is a change of location. And that is how they do it to avoid the dry and arid conditions. And some examples of C4 photosynthesis plants are corn, and some examples of CAM photosynthesis are pineapples.